and we'll move on to the next talk. So the next speaker is Christos Mitsanis from the University of Queensland. And Christos is going to talk to us about a little bit of work you had introduced earlier when Nicole referred to a poster that was pulled together by the team, but leveraging Parsimonious model PSoup for cross-scale plant modeling. And we'll be looking at the branching case study. So Christos, over to you.
Yeah, thank you, Mark. Hello, everyone. My name is Christos. I'm a PhD student in Plant Success Center, the University of Queensland. And today I will be talking about how we integrated molecular networks into crop growth models to do a cross-scale plant modeling by using PSoup, and particularly a case study on plant branching. Oops, yeah. My main aim of my PhD is to enhance genotype-to-phenotype prediction capability by introducing molecular networks into crop growth models. And currently, the genotype-to-phenotype models use the infinitesimal model to connect directly the genes and the codes of these genes into the phenotypic traits of or in organ scale of crop growth models. However, we know that instead of the infinitesimal model, we have there a trait network in between, and that have many interactions that we cannot capture with the infinitesimal model. And if we want to create this trait network and model it, we need to add some other scales in between. So first we need to have gene networks, and then after the gene networks are the metabolomic networks. So the modeling techniques that we have to model these scales, they're either ODEs, which are they're quite complex, and when we want to model gene networks and scale the map, that creates a bit of a problem because due to many coefficients and the parameterization of these. And then also there is a lot of interactions in between scales, and that is not well captured by the current modeling techniques. And lastly, also is not well captured how environment is affecting, for example, a specific gene network or metabolomic network. So these are issues that we'll try to solve with this PSoup package that I'll talk later on. And we're going to do that for a case study in suit branching. And why we chose the suit branching is because the underlying branching network that defines branching in different species is conserved across these species, which don't look alike. And it's also a heritable trait highly affected by G by E by M. So here we see the 4 stages that I did in order to create this cross-scale model and integrate the molecular network. And I will start with the first one, which is the creation of the network. Yeah. So after looking into prior knowledge, we ended up doing this network that you can see here. The different colors are indicating different pathways. And in this network, you will observe that we'll not only have genes or hormones or proteins, but we have also biological processes. We have also environmental cues like low infrared and sucrose, which is defined by environment. And here we model how all these are connecting and affecting each other, and then in the end, how they affect sustained growth, which essentially is the branching that we want to study. However, to create this, we used studies for molecular studies, for studies in agronomy, so the data are quite diverse. So how we gonna normalize all this, and we're gonna do that by using PSoup. So PSoup is a package developed by Nicole Fortuna. Probably you have talked to her in her poster. And what PSoup generally does is that it translates a network into algebraic formulas following some algebraic rules. And these algebraic rules are designed in a way to be able to capture how perturbations from the wild type are happening. Something like that. So to explain a bit more how it works is that when when everything is 1, then that means that the network is of the wild type and nothing is changed, so it's in a steady state. So if you have everything 1, then 1 equals 1 and nothing is changed, as you can see there. But then when you do one perturbation, in that case, as you can see here, we change, for example, the max to input of the gene, and we then all the formulas start changing, and then we wait till the system finds again the steady state, and then at some point it will find, and then we then go and care how this sustained growth is in comparison with the wild type. So in that case, when the max 2 is 0.6, sustained growth would be 1.5, and we always compare that to the wild type, which you can see here with a rate line, which is a baseline. And so in our case, we had as input our network into PSoup, and then it gives us as an output all the formulas that defines this network. And for simulations, we did 286 simulations. Well, 114 of them, they were unique. And when I talk about simulations, I mean they were different. For example, mutants. There were double, triple mutants. There were also mutants plus the exogenous supply of a hormone and others. And these all came from 51 branching studies in different species. Sorry. And then we went to validate, and we used 2 methods. So first, I used beam validation, which just beam validation shows us if a perturbation, for example, BRCA1 mutant, is higher or lower than the wild type. And then we found quite high accuracy, but then we wanted to see how different mutants are branching in comparison with other mutants. So that's why we used this Kendall-Tau distance. And how this Kendall-Tau distance works, it actually, Kendall-Tau distance says to us how 2 ranking orders are, how much equal or unequal they are. So for example, if we have 3 genotypes, and here we have different branching numbers in different genotypes, they give us a ranking, and then we do the simulation, we have to take a ranking, and then we compare how similar they or dissimilar they are. In that case, they're exactly similar, so the Kendall-Tau distance will be 0. And we did that for the 51 the papers that I talked before. And here, what you see in this graph, in the x-axis, there are the 51 papers, and in the y-axis, the normalized Kendall-Tau distance. 0 means that they're exactly the same. They simulate in biological ranking order, and 1, they are totally different. And on the top, you can see how many genotypes there were in each experiment. So, for example, how many mutants or treatments that I talked before. So here we see there were just 2. So the chance to be randomly correct with a random network is quite high, 50%. But as we move to 16 genotypes that we had in one paper, then we have very slight chances for these to be correct. So that was quite encouraging results. And I have to mention that these papers were that was giving a bit dissimilarity. There were papers that had, but our network was not so, let's say, robust because there were not so many information about it. And now I'm going to talk about how we connected with APSIM and why we used specifically APSIM-Sorghum. We use that because APSIM-Sorghum includes dynamic tillering, and also APSIM-Sorghum has been used from Mark Cooper and Owen Powell for the crop growth model whole genome prediction algorithm. And a bit background of how APSIM was calculating the total tiller number. So APSIM was calculating the total tiller number in an empirical way. So in this linear equation, which is the propensity of tillering, which is the intercept, which propensity of tillering is affected by the hormonal network that we're looking before, plus the slope times the supply-demand ratio. And we wanted to move away from this because we see that in reality, we don't have this linear, it doesn't behave linearly. So here we see a bit in more details how the tillering model works. And here you have with green the environmental effect, and then with the genotypic is with gray, and then you have different states and different rates. And then at some point here, we calculate the total tiller number, which is defined by the supply-demand ratio, which this is defined by the environment. And what we did is that then, of course, we calculate the supply-demand ratio still from APSIM, and then this supply-demand ratio we translate it into sucrose, which then goes into our model, and we define a genotype that we would like to define that give us a sustained growth, and that is translated into the potential total tiller number. Here we did a lot of scaling to translate things from APSIM to PSoup network, and then all of the others are still the same. And still we have some other genotypic effect. However, we don't know the underlying network, but we can also add it if we have more information about it. And what we managed to do by doing that is that we move away from this empirical linear calculation. And here you can see that we're able to see the different genotypes that here we define in PSoup. They behave differently in different supply-demand ratio. And for example, if we see this genotype, it starts from very low, and it ends up being very high and very high supply-demand index, which means in environments with high resources. So our next step is to validate this model with field data of sorghum, which David Jordan does an experiment with different genotypes and in different environments. And then if that's the case, we're going to create an in silico experiment to see what genes are affecting branching in different environments. And finally, to collaborate with Mark Cooper team and Owen Powell to try to put that model as a hierarchical model into the whole genome prediction crop growth model algorithm. And in conclusion, so here we saw that maybe high complexity can potentially be modeled in a simple way and still remain the predictive power. We saw that the topology and direction is quite important and maybe sufficient for capturing complexity and for connections across scales. And PSoup can successfully encapsulate these characteristics. And finally, that maybe integrate PSoup with crop growth model and has the predictive accuracy across different environments. So, and finally, I would like to acknowledge my supervisors, the breeding team, my Cooper own, and APSIM development team helping me, connecting it, and generally the ARC Plant Success people. Thank you very much.
Thank you very much, Christos. I think we can take one question if anybody would like to ask one. Anybody who's willing to accept the throw of the microphone? There you go. Oh, sorry, Fred.
Okay.
You get to do the final comment, so.
Thank you, Christos, very much. I wanted to ask you how sensitive is your model to the various parameters you're tuning, or how big are the differences between different genotypes for these parameters? Are all of them worth estimating, or some of them are less important? Thank you.
Yes, thank you. You mean no parameters, but you mean the genes, the genes, right? Yeah, exactly. Some are not. Yeah, the APSIM parameters, we don't change them at the moment. We do scaling. So, however, some of the genes in our network or nodes, let's say, are not sensitive, so they don't change much things in different environments. So when I was talking about the factorial, we're going to do first a sensitivity analysis to see which are worthwhile to change rather than just go and change everything, because there are so many nodes and that will lead to many combinations. Yeah.
Okay, Fred, you get your chance.
No,
Go
it's a
for
very
it.
simple question. I mean, you showed your PSoup number of tillers and the observed number of tillers. Can you bring that slide back? Yeah, no, you go back. You were there already. This one. Yeah. So if you look at the axis, I mean, observed about 5 as maximum, and you simulate about 18. So what's going on?
Oh yeah, that's because I pushed here the supply-demand index, and here is till 20. So if you see here till 20, it's kind.
But they can produce 20. I don't know the crop that well.
Sorghum can produce
so,
up to
yeah, we have
more tillers,
in the
yeah.
diversity panel that David Jordan does, it has some genotypes that they behave in that way. Yeah, so hypothetically can happen.
Yeah, no, that's clear. That's sort of earlier experiment that is simulating the diversity panel. So thank you very much, Christos.
Yep.
Very good.
My pleasure.